And thank you for coming. It's a great honor for me to be here, so I'm very grateful to the organizers for inviting me. I had not been in Argentina before, so I used the first week preceding this meeting to explore the country and was fascinated by Patagonia. But I have to say that my heart has been with Argentina for many years because I'm a big football fan. I'm a big admirer of Argentinian football and of the Argentinian football heroes, but probably to the dismay of all football fans in the audience, and particularly of my friend and colleague Damian Refojo, my favorite Argentinian football teams are the national teams of 1990, 2006, and 2010. I apologize for this, but you must understand that my patriotism has won over my admiration for the better players. With this said, I continue in the football terms just by saying that the project I will present today is a long-standing project, and it's thanks to the great patience of the Max Planck Society that we have been able to continue with it. To continue with it for a long time. I would just like to state a few names of people who have worked on the project, and then show that slide later on. So the work that I will present was mainly done by Stéphane Jamin and Frédéric Varocco in my lab, and please allow me this, and many collaborators whose names I will mention towards the end. Thematically, the project that I will present concerns synapses. Synapses are the main information processing units in the brain. We've heard about them all along the talks preceding my talk, but I would like to dwell on that issue for a little longer and describe to you a few facets of synapses that I find particularly interesting and that will be relevant for the remainder of the talk. So first of all, synapses are extremely complex molecular cell biological machines. And this becomes immediately obvious when you consider what synapses do. So down here on the right-hand side, you see an image of a synapse as sketched by an artist, which consists of a presynaptic bouton and a postsynaptic compartment. The presynaptic compartment of a synapse is specialized to respond to membrane depolarizations and concomitant influx of calcium by fusing synaptic vesicles that are filled with neurotransmitter and that then release the transmitter content into the synaptic cleft. The transmitter is sensed on the postsynaptic side, typically by ligand-gated ion channels, as illustrated here, that bind the neurotransmitter in open pores in response to ligand binding, changing the physiological state of the responding part of the synapse. And underlying these processes is a very complex molecular set of processes which involves thousands of proteins that are dedicated both to the presynaptic machinery involved in transmitter release and the postsynaptic machinery sensing the released neurotransmitter. So this is an aspect of complexity to which I will return during the talk. And the second important aspect of complexity regarding synapses is that we have a lot of them in our brain. Each of the 10 to 11 neurons in our brain make on average about 1,000 to 10,000 synapses, leading to a network of 10 to the 14 and to 10 to the 15 synapses. And here the question arises how order is achieved in the face of such complexity. And to understand how these two levels of complexity are achieved and dealt with as synapses are formed in the brain, we have to try to understand how synapses are made in the first place. We usually define two operationally characterized steps of synaptogenesis. One is the initial contact formation, during which an outgrowing sending process of a synapse has to recognize the right target cell in order to form a nascent contact. And this nascent contact will then in the course of a maturation process involving protein and membrane sorting processes as well as transport processes of different kinds will mature into a full-blown synapse as was illustrated before. And both of these synaptogenic processes are thought to be controlled by cell adhesion proteins. This is illustrated down here on the left-hand side. The initiation process of synaptogenesis, the cell type recognition phase of synaptogenesis, is thought to be mediated by synaptic cell adhesion proteins that allow specific recognition between the two contacting cells by a lock and key principle, which has been proposed many years ago. But in the mammalian system, at least, we don't know of any such process that would define cell type to cell type specific synaptic interaction. So the search is still out. Lots of candidates have been discussed, including the neuraligands, about which I will talk in the course of the talk today. But I think this process, as illustrated here, still requires facts to be proven. Like the initiation phase of synaptogenesis, the synapse maturation phase is also thought to be regulated by synaptic adhesion proteins. In this regard, these adhesion proteins form transsynaptic contacts at an already existing synaptic contact, as illustrated down here, and then in turn bind intracellular scaffold proteins, which again in turn recruit necessary proteins, in this case illustrated to the postsynaptic site, for example, neurotransmitter receptors. And the neuraligands, about which I will talk further today, have been discussed to be involved in both of these synaptogenic processes. And I will briefly describe what neuraligands are. These are type 1 transmembrane proteins with a large extracellular adhesion protein-like domain that is related, distantly related to serine esterases, and a single intracellular C-terminus that ends in a short motif by which these molecules can bind to PDC domain containing scaffold proteins. Neuraligands are localized on postsynaptic sites of synapses and bind transsynaptically to presynaptic proteins called neurexins. These have different adhesion type extracellular domains, and again a single transmembrane domain and a C-terminus that ends in PDC domain binding motifs. Mice have 4 neuroligand genes, neuroligand 1, 2, 3, and 4. Men have 5. Actually, guys have 5. Women have only 4. It's probably a disadvantage to have 5, my wife says. Neuroligand 1 and neuroligand 2 are specifically localized to glutamatergic and GABAergic synapses respectively, and I will talk about this again later on. Neuroligand 3 is present at both types of synapses, and neuroligand 4 has not been characterized as well as the other 3 proteins. What is particularly interesting to me, and hopefully also to you, is that neuroligand genes are mutated in certain forms of monogenic heritable autism. And this involves neuroligand 3 and neuroligand 4X. And again, this will be a topic to which I will return towards the end of the talk. Neuraligands have been studied by many different ways. Prior to our entrance into the field, people have studied neuroligand function by overexpressing them or knocking them down in neurons. And these experiments led to the notion that neuraligands actually might be important for this target cell recognition initiation phase of synaptogenesis. We have followed a mouse genetic approach to study neuraligands. We have knocked them out in various different combinations and found, and this is probably the first take-home message, that they are not required at all for the initiation phase of synaptogenesis. In fact, you can delete all neuraligands from a mouse brain and you still make synapses at normal numbers. What you cause, though, is a dramatic defect in synapse function. And how this comes about, I would like to explain by using neuroligand 2 as an example. Neuroligand 2 is, as far as I see it, probably the best characterized neuroligand in terms of the mechanism by which it operates. And it is also important because it will, as you will see later, become the role model for the autism related to neuroligand 4. What I'm showing you here is an immunogold labeling of a P-phase freeze fracture specimen of an inhibitory synapse labeled with antibodies to neuroligand 2 coupled to large gold particles and with antibodies to GABA A receptors indicated by the small gold particles. And you see that these are intermingled at this synaptic site, indicating and supporting prior evidence indicating that neuroligand 2 is very specifically localized only to GABAergic inhibitory synapses. we and others characterized the molecular basis of how neuroligands are operating at these synapses in quite some detail, and in order to save some time, I just give you the summary of these data. Neuroligin 2, as all the other neuroligands, binds to presynaptic neurexins. We think that the neurexins are instructive in targeting neuroligands to the nascent synapse. As the neuroligands are recruited to nascent synapses, they bind to two important scaffold proteins called colibistin and gefferin. And the binding to these two scaffold molecules recruits these scaffold molecules to the growing synapse. And these scaffold molecules, in turn, can associate into larger assemblies by way of oligomerization. And in a final step, these scaffold proteins, which are known to bind various types of neurotransmitter receptors, recruit GABA A receptors and glycine receptors to inhibitory synapses. So this is a summary of a rather extended endeavor into the biochemistry of these molecules. And I think this is a very nicely acknowledged and appreciated mechanism by which these molecules operate. And I would like to show you a few pieces of evidence obtained in corresponding knockout mice supporting the notion that these molecules are really important for recruiting GABA receptors and glycine receptors to synapses. What I'm showing you here are images of wild-type neurons and neuroligin 2 knockout neurons stained for a presynaptic marker called synapsin and a postsynaptic marker called gefferin. The prior in green and the latter in red. What you're looking at here is at the surface of a neuron, and you see that pre- and postsynaptic markers nicely coincide, labeling at the light microscopic level inhibitory synaptic inputs to the surface of this cell. And you see a nice coincidence of pre- and postsynaptic labeling in the wild-type situation. In the knockout situation, you still have presynaptic terminals ending upon these neurons, but the postsynaptic sites are more or less eliminated, at least in these perisomatic synaptic profiles. And instead, what you see is that gefferin accumulates intracellularly. We quantified that in quite some detail, and all the data basically support the notion that in the absence of neuroligin 2, you still form synapses that can also be discerned at the electron microscopic level, but these synapses are postsynaptically mal-equipped. They are not containing the proper assembly of transmitter receptors, and as you will see in the next slide, correspondingly dysfunction. This is a data set obtained in cultured cells, but you see the same phenomenon in situ in more or less every brain region that you want to look at. Here you see the consequent physiological changes. This is a recording of inhibitory synaptic transmission in a brainstem nucleus that we have studied more or less for historic reasons. The reasons are actually irrelevant for today. What you see here are responses of neurons in this nucleus down here after stimulation with an extracellular electrode to GABAergic and glycinergic inputs in this case. And you see that the wild-type cells respond with very strong postsynaptic currents and the knockout cells respond with less current. This can again be quantified in quite some detail and illustrating the notion that after elimination of neuroligin 2 from these cells, and the same is true for many other cells, you mal-equip postsynapses, you don't recruit receptors to these synapses properly anymore, and as a consequence, these synapses dysfunction. We see similar effects in the dentate gyrus, and what is important to note is that the number of synapses is not changed, not even in this system here, and that there is no change in any other neurotransmitter system. So this is a very specific inhibitory synaptic defect that we are seeing here. So to summarize that briefly, I would like you to remember that neuroligands regulate the maturation, not the initial formation of synapses, by recruiting receptors to synaptic contacts via scaffold and regulatory proteins. Magucs like PSE95 in the case of neuroligin 1, which I haven't discussed today, and gefferin and colibistin in the case of neuroligin 2, as you saw previously. There are also transsynaptic effects on presynaptic maturation, but these shall not be discussed further today. In the absence of neuroligands, synapses are made, so these proteins are not important for making synapses in the first place, but they malfunction. Now I would like to turn to the autism link. I told you earlier that mutations in neuroligin 3 and neuroligin 4 genes can lead to monogenic heritable autism in human patients. To understand that, I would like to briefly introduce autism or autism spectrum disorders. This is a very complex set of diseases with rather high prevalence. About 0.6% of kids suffer from this disorder. It is characterized by 3 main symptom classes: aberrant social interactions, so these kids often cannot make eye contact, they cannot communicate properly, they cannot interpret the state of mind of their parents, for example. There is often aberrant language acquisition. In severe cases, the children even fail to speak at all. And finally, there is a set of aberrant behaviors, repetitive behaviors, hand flapping and things like this. And depending on the severity of these symptoms and the combinations of these symptoms, we differentiate between different forms of autism. What you probably know best is the Asperger type autism of also prominent patients that are featured in the media. Often also these patients have, we can call them patients, they have also very specific savant abilities which have made them very interesting also to scientists. But what is actually the downside of all this is that 60% of autism patients actually suffer from mental retardation. So the patients or the people that we see in the media are usually the exception. What is interesting for us as synaptic physiologists and also as neuroscientists and as geneticists is that autism belongs probably to the most heritable neuropsychiatric disorder known. This had already been elucidated very early on when people studied monozygotic twins and the concordance rate in the disease among monozygotic twins, which is in the range of 80 to 95%, so it's very high, but there is obviously not 100% penetrance, so there must be exogenous factors, but most of these exogenous factors are unknown. And in addition, it has been known for quite some time that autism-like features are associated with known genetic syndromes, for example, with Rett syndrome. But most importantly, and I've kind of said that now several times, and I will reiterate it for a final time, most important for us is that there are now several forms of monogenic heritable autism, and many of these involve mutations in genes that are involved in synapse assembly or synapse maturation. I mentioned neuroligin 3 and neuroligin 4 already, but SHANK2 and SHANK3, as well as Norexin 1, have been discovered in this context. And among these, neuroligin 4 loss of function mutations are really among the most frequent and best replicated. In fact, there are even families known in which neuroligin 4 mutations have been propagated through the generations, demonstrating that this mutation alone may be sufficient to cause autism spectrum disorder in patients. All these pieces of information led to the notion that there's a protein network that determines the maturation and function of synapses in the brain that is at the core of a major autism susceptibility pathway, and we will discuss that repetitively towards the end of the talk. We have studied neuroligin